We understand genetics today as the mathematical study of heredity and variation. It is a field defined by the precision William Bateson sought when he formally coined the term in 1905 to describe how traits move across generations. But before 1865, humanity possessed no working mechanism for inheritance. While the effects of selective breeding were visible in agriculture, the explanations relied on philosophical speculation rather than empirical data. The Greek philosopher Empedocles proposed a fluid theory, arguing that parents produced distinct bodily fluids that physically mixed during reproduction. In this model, a child's arm might be formed by the mother's fluid, while their eyes were formed by the father's. Aristotle countered with his reproductive blood theory. He posited that purified male blood, upon contact with female reproductive fluids, would coagulate to form the embryo. These ancient models survived for millennia because they felt intuitively correct. They relied on qualitative deduction, observing the world and working backward to a logical cause, rather than isolating variables to measure biological outcomes. By the 17th century, early microscopes led to the preformation theory. Observers convinced themselves they saw a tiny, fully formed human, a homunculus, nestled inside a sperm cell. Under this framework, development was a matter of scale. The creature simply expanded in size until birth. Charles Darwin also struggled to find a mechanism for heredity. In 1868, attempting to support his theory of evolution, he proposed a concept called pangenesis. Darwin hypothesized that every organ in the body generated minute particles called pangenes. These particles supposedly traveled through the body to collect in the gametes, passing a blended blueprint of every individual body part to the offspring. Pangenesis and preformation both relied on descriptions of unseen processes. Because you cannot count a fluid or measure an invisible particle, these models remained speculative and untestable. Botanists in the 18th and 19th centuries, including Knight, Goss, and Kölreuter, attempted to find a physical mechanism for inheritance by crossing different plant varieties. Their experimental designs failed because they attempted to track the traits of the entire plant at once, crossing complex hybrids. They were simultaneously observing stem height, leaf shape, and flower color. This approach produced an unreadable mess of intermediate blends. Without a mathematical framework to isolate individual variables, it was impossible to see the rules governing the data. The barrier to discovery was not a lack of effort. These hybridists were performing the labor, but they were missing the statistical tools required to interpret biological results. To find mathematical laws in biology, a researcher must first control the transmission mechanism. This requires a distinction between asexual and sexual reproduction. Asexual reproduction is monoparental. A single organism undergoes mitosis to produce offspring known as ramets. A group of ramets forms a clone, where every member shares an identical genotype with the parent. Because there is no recombination, there is no variation for a geneticist to track. Sexual reproduction is biparental. When male and female gametes fuse together, they create true offspring. This fusion creates genetic recombination. The resulting offspring possess a unique genetic signature, distinctly different from either individual parent. Variation is the prerequisite for genetic analysis. By utilizing sexual reproduction, Mendel had an engine that would move specific traits across generations in observable ways. At the Augustinian monastery in Brunn, Gregor Mendel executed a research program involving more than 10,000 specific plant crosses over the course of 14 years. This volume of data was made possible by his choice of model organism, the common pea plant, Pisum sativum. Because the pea is an annual plant with a short life cycle, Mendel could observe multiple generations of offspring in a single year. Furthermore, pea plants naturally self-pollinate within their closed petals. This ensured his starting plants were homozygous, or pure breeding, providing a stable genetic baseline. By utilizing an organism with high reproductive speed and genetic purity, Mendel could generate a sample size large enough to reveal statistical patterns. To control his crosses, Mendel used a technique called emasculation. He manually removed the male stamen from a flower before it matured, preventing self-pollination and allowing him to introduce pollen from a specific, chosen parent. With reproduction controlled, Mendel narrowed his focus. Unlike earlier hybridists who studied the whole plant, he tracked only one single character at a time. He selected 7 specific traits: seed shape, cotyledon color, flower color, pod shape, pod color, flower position, and plant height. These were binary traits. A seed was either round or wrinkled, a pod was either green or yellow. There were no intermediate blends to confuse the results. By selecting mutually exclusive traits, Mendel eliminated the ambiguity that had stalled previous research. He had reduced biological complexity to discrete data points. This allowed him to apply the mathematical rigor necessary to uncover the laws governing life. By tracking binary traits across thousands of crosses, Mendel generated a volume of data that revealed consistent mathematical patterns. When crossing purebred parents, the first generation of offspring showed only the dominant trait. But in the second generation, the hidden recessive trait reappeared in a predictable ratio, 3:1. In 1865, Mendel presented Experiments on Plant Hybridization to the Natural History Society in Brunn, providing the first statistical proof for the laws of inheritance. The work was ignored. The scientific community was preoccupied with Darwinian evolution and lacked the statistical training to interpret the significance of Mendel's ratios. The history of the 1865 paper shows that even mathematically sound data remains invisible if the audience lacks the framework to interpret it. Seeking recognition, Mendel contacted the botanist Carl von Nageli. Nageli was skeptical of the pea plant data and urged Mendel to replicate his results using hawkweed. Mendel complied, but the hawkweed experiments failed to produce the 3:1 ratios. The results were chaotic, leading Mendel to doubt the validity of his own laws. Neither man realized that hawkweed reproduces through apomixis, a process where the plant produces viable seeds asexually without fertilization. Because apomixis is asexual, the seeds are exact genetic clones of the parent. There is no recombination, making the laws of inheritance impossible to observe. An unknown biological variable, apomixis, nearly suppressed one of the most significant discoveries in scientific history. Mendel died in 1884 without receiving credit for his work. In 1900, 3 botanists, De Vries, Correns, and von Schermach, independently conducted their own inheritance experiments. As they prepared to publish their conclusions, they discovered that an unknown monk had reached the same results 35 years earlier. This rediscovery validated Mendel's laws and established the foundation for modern genetics. It confirmed Gregor Mendel's legacy as the father of genetics. His work demonstrates that scientific progress often requires looking past biology and using the rigid logic of mathematics to solve the mysteries of life.